tell you some basic about the molecular phylogeny. So I will start with the question, then what is a phylogeny? So the phylogeny is a scientific discipline that addresses the history of evolutionary relationship between the organisms. Then what we need to do the phylogeny. The phylogeny can be done with the various features, with the various characters, and I have highlighted some of the features that we use to do to reconstruct the phylogeny. So the reconstruction of the phylogenetic tree is based on either morphological features, biochemical, structural characters, or the molecular character. These are the some that I have highlighted that we could use to reconstruct the molecular phylogenetic tree. So let's imagine if we want to do the phylogenetic tree with the morphological features, let's say like Acedia or Soralia in lichens or the Pothecia. So you could just code with the 0, 1, 0, 1 and then have a data matrix and then do the phylogenetic tree. So this is the one way for doing the phylogenetic tree. So everything phylogenetic tree is not the molecular phylogenetic tree. So you should be aware what the data has been used to generate the phylogenetic tree or phylogram. So it is very important for us to know whether the phylogram is based on the morphological features, biochemical features, the structural features, anatomical features. What are the features that have been used to reconstruct the phylogenetic tree? So nowadays, so the reconstruction of phylogenetic tree or the tree topology relies more on the molecular characters, that is DNA and protein sequences. Why the molecular characters? Why we don't rely more on the morphological features? As we all know that the molecular characters, DNA, protein sequences encodes the morphological or biochemical features. Because of that, we trust more on the topology, on the tree topology based on the molecular features. So this is the one thing. Second thing is that for the reconstruction of a phylogenetic tree, so we have to have some criteria for the selection of the molecular markers. So not every marker is suitable for the phylogenetic tree. So let's imagine if we want to reconstruct phylogenetic tree across the eukaryote. Where you have algae, you have fungi, you have the animals. And if we select a molecular markers of the chloroplast genomic region, let's say the rubiscose or RBCL, so it won't work to establish the phylogenetic relation across the eukaryotic tree, because it is very important that all the organisms should have the marker that you have selected. If we select RBCL, it will be present in algae, it will be present in higher plants, but it will be absent from the fungi, or it will be absent in the animals. So it's not suitable to reconstruct the phylogenetic relation across the eukaryotic tree of life. So we have to have in our mind that the marker that we select should present in the organism that we want to establish the phylogenetic relation. One criteria. Then for that, what is most suitable is the nuclear marker. Nuclear marker is present in every organism, including the prokaryotes. So if you want to do the nuclear marker or the mitochondrial markers for, let's say, for the eukaryotes, are the best marker. Or if we want to just establish the phylogenetic relation among plants or among algae, then we could use the chloroplastic region. So this is the one criteria that we have to follow. Second criteria is called selection of molecular marker is based on our objectives. Let's imagine if we want to establish the phylogenetic relation between species. So we have to select a marker that should have the sufficient variation in the region. And if we want to establish the phylogenetic relation in the higher taxonomic level, let's say families, order, etc., in that case we have to select a marker that are more conserved region, because the more variable markers will be very difficult to align. So for phylogeny, for the species level, it is important that the markers should have sufficient variation to discriminate the species. It is the second criteria that we should follow to reconstruct, to select the molecular marker. So I have given an example here. If you see, this is the short subunit, this is the large subunit, and in between the two subunits, this is the ITS region that joins both subunits in the nuclear genome. So this is the ITS1, ITS2, and 5.8. Yes, so these are the region. This is the suitable for the phylogenetic for species discrimination in lichens, in fungi, but it's not suitable for species discrimination, let's say, in higher plant. So the genetic variation, it depends on the divergence time of the species. If the species is diverged more recently, the same marker will not have sufficient genetic variation to species discrimination. So it's very important that the time is important to accumulate the genetic variation. Let's say in lichens or fungi, it has been considered today the ITS is a universal DNA barcode marker, but it's not suitable for all the groups, even in lichen. Let's say the group that has evolved recently, some group of Usnea, some group of Bryoria, the ITS is not suitable for species discrimination. This is because that the time spent, the time is less for the species divergence. If the species are diverse more than, let's say, 1 million years ago since now, then the ITS is sufficient. But if it has been diverse, let's say 50,000 years or 100,000 years, then the ITS is not suitable. This is just to let you know. This is the one criteria. Second criteria. And third criteria, today we know these are the markers. It's the SSU, LSU, ITS, RPB1, etc. These are the commonly used markers in lichens to establish a phylogenetic relation, and they work well. Imagine if you want to select a new markers, or when the first time the ITS or LSU has been selected to establish the phylogenetic relation. How do we know whether these markers is suitable, has the enough phylogenetic potential to establish phylogenetic relation or not? So this is the second criteria that we should follow. But for the lichens or for the fungi, today is not a problem. We have several markers that we know that works perfectly. But just I wanted to tell you an example. For that, what we need to know, we should calculate the phylogenetic informativeness of particular markers to know whether these markers have enough phylogenetic potential to establish the phylogenetic relation or not. So for what you need to do, there are several programs. I leave a reference here for you in the New Phytologist that we had published a paper in 2015. In that, what we did, we calculated the phylogenetic informativeness or phylogenetic information of the sex markers that is commonly used in lichens to establish the phylogenetic relation. So we use the familiacy here, data set, and then calculate it. If you see in the graph here, in this graph here, this is the nuclear LSU and MTSSU is the least informative comparing these four markers. So here in this site, you have the phylogenetic informativeness per site, and this is the divergence time of the species. If you look at from 0 till 150 million years ago, the divergence time of the species, and if we have a look, where is the optimum? Where is the optimum and which is the best marker? In this case, the TSR1 was resulted as the potential marker to establish phylogenetic relation. You see it starting from 0 till the species that have diverged more or less 80 or 100 million years ago. And if we want to compare the two markers, that comes very closely. Let's say the ITS and RPB1, here both are here. So then how we do know which one of the two is the best marker for our group? So then we have to check the divergence time of the species. If you look at here, so this is the RPB1 and this is the ITS. So ITS is more suitable to establish phylogenetic relation in the species that have diverged more or less 40 million years ago. 0 till 40 million years ago is more suitable, or you could say until here. But the RPV1 is suitable, more suitable for the species that have diverged more or less the group have diverged until 100 million years ago. It means this is more suitable for high-level phylogeny, and ITS is more suitable for low-level phylogeny. So this is just, I wanted to tell you, if you want to calculate the phylogenetic informativeness of a particular marker, existing marker, to know if in your group it's working or not. So with the phylogeny, what we could do, we could establish the phylogenetic relations using 1, 2, 3, 4 until 10 or 12 or 15 markers. But the phylogeny, it doesn't allow to analyze all the genes that are available in the genomes. For that, what we need a phylogenomic inference, phylogenomic approach. So in very short, I will explain you what is a phylogenomic inference. Today, you must have read several papers on phylogenomic inference. Phylogenomic allows to analyze all the genes in the genome. I will repeat, the phylogenomic inference allows to analyze all the genetic information of a species that are encoded in the genes. So what we need for that, so first you should sequence the entire genome through the next generation sequencing, and the next step after the sequencing is the assembly and filtering, etc. I don't want to go in detail, but the phylogeny, this is the important, is once the genomic data has been established or has been developed, the next step is we need to do the homology or the orthology assessment. So what is the homology or orthology assessment? Homology means there are two genes, at least two genes present in the genome. Orthology means the single genes are present in the genome, or the single copy, that we say the single copy gene. So for the to establish the phylogenetic relation, what we need only the orthologous gene, not the homologous gene, because the homologous gene, two copies are present, and it could give you the different phylogenetic relation for gene. So we discard all the homologous gene, and then we concentrate only on the orthologous gene. And after the orthologous gene selection, then we do calculate the phylogenetic informativeness among all the orthologous genes, and then only do the phylogeny that have the phylogenetic potential. You could also do all the orthologous genes that are available in the genome. So then the next step is then the alignment. You could align separately all the genes and then make the concatenation, make the combined data set, and then this will come in a supermatrix, and you could do the phylogeny of the supermatrix, let's say the 1,000, 2,000, etc. the genes, and this will be the tree. This is the phylogenetic, the gene tree. And then simultaneously, what we do a species tree. For a species tree, what we need to do, we have to analyze all the genes independently. I mean, gene 1, gene 2, gene 3, gene 4, etc. And then once we have the tree of all the genes, then we do the species tree. Species tree means the most common topology. Then we end up with one tree that has the most common topology among the thousands of the tree that has been generated with the single gene data set. So we end up gene tree and species tree. That way we could analyze all the genetic information of a species with the phylogenomic inference. I will leave a reference again here. If you want to go in detail, in this study, we had analyzed 2,556 single copy orthologous protein coding gene to establish the phylogenetic relations using the phylogenomic approach. So these were the basics that I wanted to tell you before going to the application of the molecular phylogeny. Remember, I will only concentrate on the molecular phylogeny. Phylogeny can be with the morphological, chemical, biochemical, atomical data, but I will concentrate only on the phylogenetic tree based on the molecular character, that is molecular phylogeny. So I will select for today a model system, that is the Parmeniaceae. So then why I selected the Parmeniaceae as a model system for showing the application? For the two reasons. The one, so this is the one of the largest families in Lichenized Escumicota and represents about 15% of the total diversity of Lichenized Escumicids, and it is widely spread. This means it is distributed along the world, including Antarctica, that allow us to address the question related with the biogeographic distribution. And it has vast morphological diversity that allow us to answer the question about the morphological plasticity. And it has the vast chemical variation. And this also allows us to know about the chemical evolution. So this is the reason that I have selected the Parmeniaceae as a model system. And another is, this is one of the most studied families in lichens and also the most common family. It is found everywhere. It is very common. So this is the two reasons that I selected Parmeniaceae as a model system for showing some of the application, not all. I will show you some of the application. So in my outline, I will show you some of the application of the phylogeny, the first resolving evolutionary relationship, how the molecular phylogeny have been helpful in Parmeniaceae to answer the long-lasting questions in the Parmeniaceae. Then I will go for the species and genus circumscription. Then I will show some example divergence time estimates, and then ancestral range reconstruction, how the molecular phylogeny had been helpful for that, and then I will end up with the general conclusion. So starting with the resolving phylogenetic relationship. In the Parmeniaceae, the first molecular studies was appeared in 1998, and thus the molecular phylogeny was based on a single molecular marker, that is the ITS region. And the two authors that published simultaneously the phylogenetic tree of the Parmeniaceae. One was Crespo and Cubero in 1998, and in the same volume, in the lichenologists, Jan-Erik Madsen and Mats Wedding presented the phylogeny of the Parmeniaceae. Both the phylogeny were based on the ITS marker. As you could see, this is just the single marker. They were lucky to establish the phylogenetic relation at the internal nodes. We'll see the 193, 100 bootstrap supports, and with the single gene, they were able to know that the so-called Neophysalia at that time were not monophyletic. They were grouped within the Xanthophamelia here with a strong support. And also the Palmotrema was not monophyletic at that time, and the Remelia reticulata was grouped within the Palmotrema. I mean, in 1990, were not monophyletic. I mean, the morphological characters were not suitable for the generic circumscription with just a single marker. Then there have been several studies. I will not go in deep for showing all, but I want to show you the other study that was published in 2007, and it was based on the 4-locus phylogeny. In that 4-locus phylogeny, and the study was based on the full family Parmeniaceae, and the author were lucky to establish the phylogenetic relation among the major groups in Parmeniaceae. And then what we do, we define 7 groups, 7 clades. That is the Parmelioid clade, Alectorioid, Coeloparmelioid, Citrarioid, that includes all the Citraria, Hippogymnoid, that includes Hippogymnia, and the Letharioids, that include the Letharia. All these clades were monophyletic. I mean, they received a strong support on the monophyly of all these major clades, and they define all these. But the phylogenetic relations or the evolutionary relations, we could say, among these clades were not supported. I mean, but remained unresolved in this study. that they did the 4 locus phylogeny. And then in the second study, what we did, we focused, this is the whole Familiaceae. You could see this, the familioid groups out of the 2,800 species, described species, 75% are grouped here in this group. And 25% of the species are in the rest of the group. Means familioids has the highest species diversity in the family Familiaceae. And then the next study was focused on this group that includes the 75% species. As I said, this is the widespread family and huge diversity, and it is very difficult for a single scientist to work on that and to collect all the species around the globe. And for that, what we did, we established at that time a consortium that was named with PARSIS, means Familiaceae Systematics, to establish phylogenetic relations in the familioids. We made a consortium, and then a total of more than 40 authors were involved in this study, and then was based on the 4 locus phylogeny, that is ITS, nuclear LSU, the mitochondrial SSU, and the one protein coding gene, that was the RPB1. And the total number of species was analyzed was 2,075 species, and representing 323 OTUs of all the markers and 760 OTUs of just ITS. ITS, we had 470 species, just ITS was analyzed in this group. And then what were the results? And you could see here in this paper, or in this work, we were able to establish the phylogenetic relations in the different groups within the familioid clades. Means you could see we were able to establish different clades within the familioid that received a strong support. That means palmotremoid clades that included Palmotrema, Cainoparmelia, Flavoparmelia, Astroparmelina, Punctilia, Cainoparmelia, and also Nisolachia and Flavoparmelia. Then we had another clade, that is Xanthoparmelia clade, includes all the Xanthoparmelia species. And then we had Citralia clade, only Citralia was included. Then Parmelina clade. In Parmelina, we had Remototrachina, Volvotrix, Parmelina milocrova. We had Hypotrichina clade here, also strongly supported. Then we had the Hypotrichina. Then we had even Neostrum, the old name, but now all are synonyms. So we had all this, and then Hypotrichina. Then we had the Parmelia clade that included the Parmelia, Relicina, and then Parmelia substructum. We had the Melanohalia clade that included Melanohalia, Melanolexia, and Montanellia was on the groups. You could see we had established the phylogenetic relations within the palmeloid clades, and most of the genera received the strong support for the phylogenetic relation. In Palmotremoid, Dina, Epitrichina, in all the clades we have almost support. But the phylogenetic relations between these clades were not supported. I mean, it remains unresolved in these groups. And then another study was focused on the remaining 25% species of the Familiaceae. In this, we continue our consortium. This was PARSIS second, or PARSIS Familiaceae Systematic, that was established in 2014. Sorry, and the paper was finally published in 2015, and the first was established in 2008, and the paper was published in 2010. In this, more than 60 authors were involved, representing 40 countries. I mean, we were able to join different groups working on that, and we mainly focused in this group, but also on the palmeloid clades, and we were more interested to include almost all representatives of the Familiaceae genera. This was the main focus, main objective. And we also increased 2 markers, that was MCM7 and TSR1. The total number of species we analyzed 274, representing 72 out of 80 accepted genera in this family. So our main focus was to know whether all the genera is described in combination monophyletic and what was their phylogenetic relation, etc. And then it was analyzed using the different approach in the phylogeny, and then we reconstructed the ancestral state, divergence time estimate, and then we did also the phylogenetic informativeness using phylogenetic design, etc. And this is the results that you could see in this group. Again, we had a strong support with the 6 markers in all these major groups, and we were able to establish new groups that was Anzioid and Usnioid clades. Here the Ansia is included and Pinopamilia included, Usnia is alone. And also we were able to establish the sister group of the Pameliaceae, that is Protoparmelia sensu-stricto, with the sister group, and also the sister group of the Pameliaceae, that is the Gypsoplacaceae. So our main focus was to establish the phylogenetic relation as the higher level, not the low level phylogeny, but more on the external level. So it was well resolved in all the clades, but in this case again the phylogenetic relation in some of the clades remained unresolved. We could see, I have highlighted here, we really don't know whether Pamelioides is the sister group. We don't know. We were unable to establish the phylogenetic relation of the Pamelioides. Neither we don't know the Gymnoid clades forms a sister relation with the Pamelioides. Here again, we have no information on the phylogenetic relation among these Citruroid, Alectoid, Ansioid, and Usnioid clades. We don't know who is sister to what. So for that, what we did, so we thought there could be 2 reasons. We were unable with the 6 markers, more than about 300 species analyzed representing all the genera. We were unable to establish phylogenetic relation. Could be 2 reasons. One, that we had not enough genetic information to establish phylogenetic relations among the clades. This is one of the reasons. I mean, we had a very small portion of the genome analyzed in the data. And another reason could be we have not the sufficient species to establish the phylogenetic relation. So we had already analyzed more species in Pamelioides, and here also are 300 species representing all the genera. So then in the next step, what we did, we thought, why not we should try to analyze first all the genetic information of the species and then see whether we were able to establish the phylogenetic relation or not. And if with the all genetic information we were unable to establish the phylogenetic relation of a group, could be that the sister group of that species might have lost in the evolution of that family. This could be one of the explanations. So in the next project, as I explained earlier, what we did, we applied the phylogenomic approach. In this, we sequenced 50 genomes representing different genera of the family, across the family, and also we included genomes of the other families, not only just the Pameliaceae, but we also included the other groups. I have not added here, but mostly I will show you here only the Pameliaceae. And then what we did after the genome sequencing, we assembled, we annotate all the genomes, we did the orthology search of all the genomes, and finally we end up with the 2,556 single copy protein coding orthologous genes. And this was analyzed in the 2 phylogenetic framework. One was the gene tree, as I explained. What we did, we concatenate all these the genes, and then we run the phylogenetic analysis. It ran on supercomputer. It took more or less a month to finish the analysis, and then was the gene tree. And then in parallel, what I put here, this is the species tree. It means all the single genes were analyzed separately, And then the species tree were calculated. There are different programs in that. We use the program called Astral to calculate, to do the species tree. And then if you see that I put the topology of both, whether they were congruent or not, and you can see most of the phylogenetic relation were congruent among the gene tree and species, except few relations that I have shown with the dotted lines. So there was incongruence with the phylogenetic relation in the Pamelinopsis and Omphilora. Here, the two groups that we were unsure which is the sister group of, this is the major, this is the Pamelioid, that you could see the major groups. So we were not sure in this whether the sister group is Pameliopsis or Omphilora, because the tree were incongruent gene and species tree. In both, we had the 100% support. You can see here with all the genetic information of the species, we found strong phylogenetic relations among all the major clades, not only just the major clades, but also at the internal and the external levels, they were strongly supported. We have 100% support at the nodes of all the phylogenetic relations. So what we could conclude with that, to establish phylogenetic relation, if we were unable to do with the classical markers, with the 10, 20 markers, then it is crucial to analyze all the genetic information before to do a conclusion. Any conclusion, it is crucial to analyze all the genetic information, and we were able to establish the phylogenetic relation in Pameliaceae, even just with 50 species representing all the genera. If you could see with the one or two species per genus were included here, not more. But the phylogenetic relation was strongly supported. So this is about how the phylogeny or the phylogenomic approach have helped to establish the phylogenetic relation in Pameliaceae. And the second is the species and the genus circumscription, how the molecular phylogeny have been helpful for that. So I will go in very brief. I will not take much time as in previous talk. So we all know the fundamental category of the biological classification of the species. There are different species concepts. You all know, so morphological species concept, biological species concept, phylogenetic species concept. So what is the morphological species concept? In brief, I have put the explanation here. A species is the smallest group of organisms that can be consistently distinguished or consistently separated by the presence or absence of the morphological features. That could be body part, shape of the lobe, shape of the size, presence, absence of the Soralia, etc., and the number of those parts. This is the morphological species concept. The biological species concept. The species is the smallest group, that means all the population of that group of smaller individuals that can reproduce with each other but cannot reproduce with other groups. That means all the population of a group, this can reproduce with each other, but this group cannot reproduce with another population or another group. This is the biological species concept. Then we have the phylogenetic species concept. This is the concept that we follow today in lichens or in fungi or in even all the organisms that we are following is the phylogenetic species concept. So what is the phylogenetic species concept? A species is a group of organisms. It's a group of organisms. I put an example, 1, 2, and 3. A group of organisms that share an ancestor. So 1, 2, and 3 share an ancestor here. And a lineage that maintain its integrity. Lineage, species 1, species 2, species 3, all these maintain its integrity with respect to other lineages, that is the species 2, through time and space. This is the time. Through time and space, it maintains its integrity. So this is the phylogenetic species concept. So I put an example here. So if we do the sampling at this time span, then we will see that species 1, species 2, species 3 all are monophyletic, because all the groups have in sufficient time to fix their leads. They received the sufficient times to fix their leads, and in other words, I can say they had sufficient time for the divergence, for the species separation. But if we sample here at this time period, we will see that the species is para or polyphyletic. That means the time has not been sufficient for that group for the separation. It's still in the process of speciation. Para and polyphyletic means the species is still in the process of speciation. The time has not come, that has not been sufficient, that alleles should fix and the species got separated independently. So this is the one example that I gave, the phylogenetic species concept. We realize more on the phylogenetic species concept today. So I will give just one example of a species concept. You know the classic example, that is Parmelia saxatilis. The species was described, we all know, as lichen saxatilis by Linnaeus in 1753. When Linnaeus described, he thought, oh, this is one species, most common species, that is the Parmelia saxatilis. In India, it's not so common, but in Europe, in North America, this is one of the most common species, is the Parmelia saxatilis. At that time when Linnaeus described, he never thought that Parmelia saxatilis could be various species. And this is one of the widespread species. I mean, the Parmelia saxatilis is bipolar species, distributed in both poles, including the Antarctica. It's also found in Antarctica and the tropics, temperate, boreal, everywhere. And when we analyzed in the phylogenetic framework, we found that the Parmelia saxatilis is not a monophyletic entity. I will give you just the last paper. There's different paper has been published. The first paper was published Morila et al. 2004, 2011, and then was in 2016 by me. And then the last was, this is published. I forget to put the reference in 2021 has been published. And if you see the Parmelia saxatilis today includes about, I put here about 5 to 7 species. This is not a single entity, but it includes 5 to 7 species. So this is thanks to the molecular phylogeny. Today we know that Parmelia saxatilis is not a single species, but it's a complex, and it includes more than 5 species in Europe and 2 in North America. Total number of species included in saxatilis is 7 today with the molecular phylogeny. So the molecular phylogeny or the DNA sequences is crucial for species discrimination in lichen. Just one example I have given here. So the different species, Parmelia May, distributed in North America. Saxatilis is still wide distributed. Serrana, mostly in Europe and North America. Nesti in Europe and North Africa is here. And Rojoy recently described in one of the region in southern Spain. So there is still species widespread. Apart from the segregation of sex species, Parmelia saxatilis structure remains widespread. It means it's still found in both poles, in Antarctica and in the Arctic regions. Another example, and I will give you for the generic circumcision, how the molecular phylogeny helped us for the generic delimitation, for the genus delimitation for the generic circumcision. So in this study, in the PAR 608, the paper published in 2010, we established some criteria for the genus circumscription. Based on this topology that we were able to establish, we established the 3 criteria for the generic circumscription. First, that the genera should be monophyletic. This is the first criteria. And the second, it should have distinct divergence time than its closely related group. And the third criteria is the lineage should be corroborated with the ecological, geographical, or the morphological feature. These were the three criteria that we proposed based on the molecular phylogeny, or in other words, we could say the molecular phylogeny helped us or allow us to propose these criteria for generic circumscription. And we were also able in this study to make the progress toward the international consensus on the generic concept in the familioid lichens. And in this study, we accepted a total of 27 genera in the familioid lichens. And then the molecular phylogeny, not only for the generic circumscription, but it's also established for help to establish other groups. Before going in other groups, I want to show the classic example of the genus concept. Xanthoparmelia today includes more than 800 species, and it includes all these genera that I have included here. More than 10 genera are included in Xanthoparmelia today. Just one genus, Xanthoparmelia. Xanthomaculina is added, Namaqua, Alambornia, Chondropsis, Carubia, Pyrapamelia, Neophysalia, Omphalodiella, and Placoparmelia. All these genera are included in a single genus, Xanthoparmelia, based on the molecular phylogeny, has helped to clarify the generic concept in the Xanthoparmelioid clades. Not only the lumping, but it also helps to the segregation of the morphologically similar groups. Let's say Hippotrachina. So one example in India, Hippotrachina, that is very common groups, and you could see here with the broad-lobed Hippotrachina that is common in India. When we put in the phylogenetic tree, both the groups but too far. I mean, they were not phylogenetic related at all. This is the Hippotrachina stricto, and look at this. This is another group, Hippotrachina, that formed the sister group with the Bulbotrex, and later it was described as the new genus, that is the Autotrophic. And then not only based on the phylogeny, but these were corroborated with the morphological features. The morphological anatomical features corroborated the segregation of the Autotrophic with the Hippotrachina, with the broad lobes, exciculum features, and the ascospores features were helped to the segregation of the Autotrophic from the Hippotrachina. And the geographic distribution, mostly the Autotrophic distribution in the Southeast Asia, and Hippotrachina in the pantropical distribution. So this is another example of the generic circumscription. It also allowed us to establish a threshold, the molecular phylogeny. So this is the dated phylogeny that I am showing today. And the paper was published in 2017. So hold the Familiaceae that we used in Parsis 2, and then we do the molecular dating of that to establish a threshold for generic circumscription. So this is another alternative approach that supplement our previous criteria for generic circumscription, that we propose 3 criteria. So this is another approach. This is just one approach we could use for the generic circumscription. No, this is an additional approach for that supplement the generic circumscription based on the molecular phylogeny and corroborated with the morphological or the chemical features. So we were able to establish a threshold for the generic segregation. That is here, I put here the genus that was diverse between 29 to 32 million years ago. They were considered genera. Less than 29 million years ago were not considered as independent genera. That comes before that threshold were synonymized in the group. So this has been used in first time in the animals. They used a lot for the dated phylogeny, established the threshold for the generic circumscription, and we used the 5% error for that time. And with this approach, we accepted a total of 70 genera in Familiaceae at that time. So this is another approach, and luckily, or we could say the most of the genera that we have described based on the phylogeny, our previous criteria that I showed you in 2010, were supported with this threshold. You could see here Pomotrema, Propermelia, Astopamelina, Punctilia, Flopamilia, Genthopamilia, Cynopamilia, Citrine, all these genera that were previously described, that were previously studied with molecular phylogeny were accepted. I mean, had this threshold. So this is that supported us. And the other groups that were not rigorously analyzed with the molecular phylogeny, here, this is the citradioid clade. And in that citradioid clade, then we had to synonymize several genera. Here, Citraria included at that time, we synonymized Allocitraria, Citrarelia, Oslocitraria, Mulposita, and Citraria, and Nephromopsis we synonymized a number of genera that has not the fall within the threshold that were these. And then we were also able to establish a threshold for the family segregation. In that case, the family segregation, the threshold fall between 100 to 112 million years ago. If the threshold were beyond the 112 million years ago, means that were not a family. So based on that, what we did here, we described a subfamily, that is to accommodate the Protoparmelia that were sister group to the rest of the Parmeliaceae, and was named with Protoparmelioideae, and then the subfamily Parmelioideae. So today there are two subfamilies in Parmeliaceae, Parmelioideae and Protoparmelioideae. So this is another approach that the molecular phylogeny is also helps to establish the threshold for the generic circumscription, also for the species circumscription. I am not showing here, but it has been helpful to calculate the genetic distances for the species circumscription. And quickly, I will show you the divergence time estimate, how the molecular phylogeny has been helpful to know when a group and when a lineage has been diverse. Looked here in the Parmelioids, in the Parmeliaceae, we were able to do the molecular dating. To do the molecular dating, it is very crucial to have a strongly supported phylogenetic tree. And then we use the program BEAST. You could go for the morphological details here and use the BEAST. We could then use the fossil record that was available. So with the data at hand, the fossil record at hand, we were able to do the molecular dating and to show that the Parmeliaceae, the family Parmeliaceae, were diverse in the Cretaceous period. I mean, 100 more or less 100 million years ago, the Parmeliaceae family was diverse, and most of the genera in the Parmeliaceae were diversified in Paleocene and the Oligocene period. Here, all the genera were diversified in that period, and most of the species were diversified in the Miocene period. So these were the results of our study, and the generic, the genus divergence, the divergence of the genera or the diversification of the genera were correlated with the mean temperature at that time. So we had also looked in the past what was the mean temperature at that time. We look at here the mean temperature in the Paleocene. It's jumped and then it starts dropping. Here it went below zero was ice age at that time. At the time of Oligocene, in the Eocene period was the first the glaciation period. At that time, most of the Parmelioid or the Parmeliaceae genera diverged when the temperature drops down. And in the ice age helped for the divergence of the genera in Parmeliaceae, and then when the temperature again rise in the Miocene period, that was helpful for the species diversification in the Parmeliaceae. So these was the two correlation of the divergence in the family Parmeliaceae. So these were also helpful from phylogeny, as can also be applied for the molecular dating of a particular species, particular group. I could just give an example of the Parmeliaceae. And it has also been applied to reconstruct the ancestral state. Ancestral state can be the morphological features, the geographical features. We could know which morphological features has been evolved first, and it also allowed to know when the species has been evolved. Earlier I showed you when the species have been evolved, and it also helped to know when a group of a species has been evolved. The ancestral state reconstruction of the geographic data. I mean, that we call the phylogeography, and it helped us the molecular phylogeny. In this group, I will give you an example of the ancestral range reconstruction of the Hippotrecina clade. This group, we used the phylogenetic tree, and then we used a program called Lagrange. We encoded the geographic distribution of a species. Look at here, we divided the globe in the different phytogeographical regions, and then encoded, let's say, Southeast Asia, North America, Southern America, Australasia. According to your interest, you should divide the globe. So in that group, we had divided the globe in Australasia, Southern and Central Africa, North America, Southern and Central America, Indian subcontinent, and Southeast Asia. These were the regions that we had divided, and then the species, accordingly, the species distribution were encoded and analyzed in using the program Lagrange. So this is the results that I am showing today. If you look at here, the Autotrophic the program said the Autotrophic was evolved in Indian subcontinent, and it evolved more or less 50 million years ago when the India was traveling, was as island, and was traveling, was somewhere in ocean at that time. And then here the Bulbotrex, one of the group of the Bulbotrex evolved in Southeast Asia, and another groups we don't know whether it evolved in Southeast Asia or Southern America. So these were some conclusion of this. So another example of the ancestral range reconstruction of this, if we used you could use the different programs here. In that case, we used another programs called BioGeo Bears, and which also used the molecular phylogeny, and then similarly we encoded the geographic distribution, and then we were interested in knowing the origin of the Milochrova and the Pamelina clade. Here Milochrova and Pamelina clade evolved more or less 37 million years ago since now, and then it's diverged in the two groups, Milochrova and Pamelina. The results where the group were evolved were not conclusive. But if you look at here, the Milochrova evolved 22 million years ago in Asia, and the Pamelina evolved more than 8 million years ago. It evolved in either Europe or Iran region. So these were some explanations to follow that help to know how the been helped for this.
Yeah.
And then we try now for
conclude, please, Diwakar.
Yeah, I am now the conclusion, so I'm running out of time. So the molecular phylogeny have helped to resolve long-lasting questions on Pameliaceae systematics, as you have seen earlier, and it has also allowed to establish criteria for species and genetic circumscription in Pameliaceae. And also today, with the knowledge we have in Pameliaceae, I can tell you with confidence that the DNA sequence-based sample identification is crucial for species circumscription, not only in lichens, not only in Pameliaceae, but in fungi in general. So we propose the genetic circumscription. There were three criteria was established. Genera should be monophyletic, should be have distinct divergence time and exclusion-related groups. The lineage should be corroborated with ecological, geographical, morphological features. And Pameliaceae evolved in mid-Cretaceous, and the major lineage diversified between Paleocene, Eocene, and Oligocene. Today, I could say with confidence, this is one of the most robust phylogenetic framework known for the lichenized fungi so far, and this can be used as model system not only in lichens but also in other groups of the ascomycete fungi. With this, I will stop talking and open the session for the questions. Thank you.
Thank you, Divakar, for a wonderful talk, and we actually wanted to hear more, but because of the time, so we happen to ask you for the conclusion. And there are few questions, and we'll take up one or two questions, very quick questions. And for example, this Sachin Singh is asking, sir, if I want to do the phylogeny of Asnia, which marker is most suitable for it? This is one first question of him. How can we select a suitable marker for phylogeny of different group? This is the second question of the same person.
Okay. With the first question, what is the suitable marker to establish the phylogenetic relation in Osmia? So as I said, the ITS is suitable for Osmia as well, but not for all the groups. In my opinion, in my experience, the best marker that we found to establish phylogenetic relation in Osmia is RPB1. The MCM7, and then the GPD, and the TSR1. So these are some markers that have helped to establish the phylogenetic relation in Osmia. RPB2 is also helpful to establish phylogenetic relation, but I will also tell that you should also include the ITS, because sometimes it's good to establish the phylogenetic relation in the internal nodes. So this is the answer of the first question. The second question was Sanju, can you repeat the second question? Sorry.
How can we select a suitable marker for phylogeny of different groups?
Of the different groups. Okay. The one criteria that I already explained, to know whether the marker that is today available. Today in lichen we know the universal marker, that are 4 or 5 markers that have been commonly used. So to know whether this is suitable for our group or not, to save the time, so what I will say you to calculate the phylogenetic informativeness. You should calculate the phylogenetic informativeness. This is the best way to know whether this is the marker is suitable for your group or not. Whether this has the phylogenetic potential to establish the phylogenetic relation or not. So I would say to calculate the phylogenetic informativeness, I use PhyloDesign, but there are other programs you could use to calculate the phylogenetic informativeness.
Yeah, I'll take up one more question, last question. Akhilesh Kumar Maurya is asking, sir, if we select a marker for 2 organisms for their species identification, but one of them have mutation in its genome, then how can we conclude the result?
I mean, one of the gene has mutation and another has not mutation.
Yeah. I mean, you have selected 2 organisms for identification.
Yeah.
But one of the organism has mutation in that particular genome. So then how we can identify, how we can conclude the result? It's a kind of tricky question.
Yeah. I'm not understanding well what you mean, but a mutation, if a region has a mutation and another has not a mutation, means that this region has the sufficient time frame to generate the mutation of a species separation. So, I mean, I will go for the region and conclude mostly on the region based on that has the mutation for the phylogeny. I mean, the region that has not the mutation, it's not helpful for that to establish phylogenetic relation. I don't know if I'm answering your question.
Okay, I mean, I'll ask him to contact you directly so that he can discuss. Okay.
Yeah.
So remaining questions I will forward to Dr. Divakar. He will answer you leisurely. And now we will move on to the next speaker. I thank Dr. Divakar for his wonderful talk and being available today to deliver his talk. Thank you, Divakar.
Thanks.
Okay, now we have with us Dr. A.E. Dulip Daniels. So, Dr. Daniels is now Associate Professor of Botany in Scott Christian College, which is an autonomous college in Nagarkoil, Tamil Nadu. He is a bryologist. He has successfully completed 3 research projects of various funding agencies, especially Ministry of Environment and Forest, DST and all. Then during his service, he has successfully guided 30 scholars for MPhil and 7 for PhD degree, and he has published more than 100 research papers and 4 books on the bioflora of Western Ghats. And among the floristic aspects he has studied, he has completed the work of bioflora of Agasthyamala Biosphere Reserve, then bioflora of Indira Gandhi National Park in Annamala and all. So, he's an active bryologist in South India. He is a founder or director of Bryology Laboratory in the Department of Botany, which is indexed in the Index Herbarium, New York Botanical Garden, United States of America. And I thank him for accepting our invitation and ready to give the invited talk, that is diversity and distribution of bryophytes in Western Ghats. So now I invite Dr. Daniels for the talk. Please, sir.
Okay. Thank you, Dr. Nayak, for the very warm words of introduction and for this nice opportunity to share with you a little of what I have done in the southern extreme of our country. Can I present?
Yes, please.
Is the screen getting displayed?
Not yet.
Not yet.
Yeah, it is coming now. Yes.
Yes.
You can make the slideshow of it.
Okay. Yes.
No, from here you select the presentation. Now we can see the different pictures. Yeah. Yes. Now you make it slideshow. Yes. Perfect.
Okay.
Go ahead.
Well, I would like to share something on the diversity and the distribution of the bryophytes, the Western Ghats.
Excuse me, can you please just put the hide slide? Please press the hide. Hide on the screen. Okay, thank you.
Okay, well. So partly based on my personal observational studies and partly based on literature. Well, this is just to tell you, this is a Braun who first used the term bryophyta, which included algae, fungi, lichens, and mosses. And later on, it included only the bryophytes, the 3 groups, the Hepatica, Anthocerata, and Mussel. Well, we all know that bryophytes were the first group of plants to migrate and colonize a terrestrial habitat from an aquatic habitat. Still, the mode of life is amphibious since they require water to complete their life cycle. Okay. In the 8 biogeographical zones, this is where I'm working. Western Ghats is a long strain of mountains range running over 1,600 kilometers from Gujarat to Kanyakumari district. And this is actually split into north and south. Earlier it used to be from Karnataka down as south, but now strictly speaking, based on the monsoon pattern received. Now strictly speaking, southern Western Ghats is south of Palghat Gap, because that's where the southern Western Ghats receives both the monsoons, both the northeast And I work in that area, and I would like to concentrate more on that. Well, this is just data. What we have so far identified, definitely these figures would change because research is going on all over. So these are tentative figures, whatever we have done all these years, and this is definitely going to change in the coming days. Well, see, as you go on based on literature and by studies, we find there is mostly distant distribution of the moss. I've given just a sample. Mosses showing distant distribution worldwide. To find Physidens palladidiversi, it says America, and next only in Asia. And in Asia, we find in Nepal, Sri Lanka, and India. When we say India, this is actually Northeast India and the Western Ghats. So there's a huge gap between when it comes. Okay. Similarly, Silchar, you could say that America, Africa, think where Papua New Guinea is, and then Asia. And within the continent, it's Malaysia and India. So similarly, you find for all the species like that, they all possess discontinuous distribution. Okay. Now, liverworts, coming to liverworts. Again, the pattern remains to be almost the same. Now, disjunct within the country, you see that the species represented were in Nicobar Islands and in the Western Ghats. There is a gap. Similarly, Africa, America, and Asia for Scripturaligia polycarpa, and Sri Lanka and India. Then you come to Drepana legernia, you could see that China, Nepal, and India. And within India, it's usually Eastern Himalaya, Northeast, and the Western Ghats. Now, moss is endemic to Indian subcontinent, showing distant distribution. See, when I say Nepal and India, in India it's actually the Western Ghats. So there is a large gap between the Himalayan and the Western Ghats. But still, there are no reports in between, in the intervening areas. So what could the reason be? Liverworts endemic to Indian subcontinent showing disjunct distribution. Again, the same. The same pattern is being represented. You could see the same. Dicranella macrospora, you see Meghalaya, the Northeast, and the Western Ghats. Philanotis leptocarpa, you could see Himalaya and the Western Ghats. Ectropothecia mannii, you could see Andaman Islands and the Western Ghats. So, this pattern seems to persist. Pterobriapsis cageliana, you could say Madhya Pradesh, Western Ghats, and Eastern Ghats. Similarly, liverworts, you could see the same pattern being exhibited. Okay. Now, the species occurrence in different habitats, because Western Ghats is blessed with almost 5 types of forests where the species occur in different habitats such as corticolous form growing on barks, folicolous growing on leaves, lignicolous growing on logs, rupicolous on rocks, saxicolous on walls and stones, and terricolous on. Even here, you could see that there is more representation. Yes? Yes. There is more representation, okay, on corticolous forms, but equally, very often they are represented, except for folicolous forms, because folicolous forms I haven't come across several mosses, just 1 or 2 mosses. But when you come to the liverworts, you can see folicolous forms, particularly in riparian forests and evergreen forests. Otherwise, they are evenly, more or less, they are distributed. We find species representation in almost all the types of habitats. Now we come to species across the different vegetation types. First is moist deciduous forest, next is degraded evergreen forest, third is evergreen forest, then we have scrub forests, tea plantations, and coffee plantations. Well, Yes, sometimes we find the species occurring in evergreen forests are also represented in tea plantations, coffee plantations. This could be because of the elevation. Particularly, bryophytes are water-loving and they are high-altitude colonizers. So this could be one reason, because these are all monoculture plantations. However, many species that occur in the evergreen forest also occur in tea plantations and coffee plantations. Similarly, you could see the same concerning hornworts and liverworts. But there is more of representation in the evergreen forest. Studies reveal that usually concerning other organisms, diversity is more in degraded forests because there you have exclusive species as well as common species. But in the case of bryophytes, studies may reveal that they are more exclusive and they prefer to grow in evergreen forests where there is higher moisture content. Well, now just let me show you some diversity because the mixed group listening to me, basically Bryophyta, is divided into three. Now today the terminology used is Marcantiophyta, Anthocerophyta and Bryophyta. In Marcantiophyta you have two, that is thalloid forms and leafy forms. Well, where do they grow? There is habitat preference also. They could grow in crevices of rocks. This is the riparian forest. You could see the fallen tree completely covered with mosses. They grow close listening to shady springs, almost submerged in water. You find them on asbestos roofs, mortar of stone and mud walls. These are quite common species. This is a very common sight in high altitude mountains where these plants, there is Bryum argenteum growing on the asbestos. And there's Plagiocasma rupestri growing on mortar of stone walls. You also have corticolous forms, polycolous forms, they call them epiphyllous, rupicolous, those growing on stones, taxicolous, growing on logs, then on soil. Then there is diversity even in the stomata. This is the stoma of a bryum taken from a bryum capsule, and this is, of course, Anthocerus sporophyte. We're coming to the gemmae. Gemmae, again, these are all most common species. Lunularia is a very common, cruciate is a very common species that you find in any high altitude range in South India. It's like a half moon with gemmae inside, but this is a complete. Marcantia polymorpha is a very common species, you should be familiar, but the difference in the gemma cup shape. Look at these. These are gemmae of various mosses and a liverwort. This is Calibrus species. Look at the variation, the diversity. This is Syntrichia species. This is Ochrobryum, and this is Cololagenia. Now features, even in the morphology, some mosses are erect, some are prostrate, and some are pendant. The stem could be simple, branched, or proliferating. That is, after the capsule is produced, from that it continues its vegetative growth, and once again, this is proliferating. Well, stem anatomy, some do not have a central strand, supposed to be believed to be the initiation, the beginning of a conductive tissue. Some do. The branching type, pinnet forms, irregular, julaceous. Even leaf shape, look at the diversity in the leaf shape of mosses. First one is Somatophyllum, next is Calympyrus, then you have Glossadelphus, Dissidens, Leucobrium, Meteoriopsis, and Bryum species again. Leaf anatomy. This is Leucobrium, Pogonatum, then Dissidens, and Syntrichia species. This is Proteaceae, that's Lepidopteroideaceae. Leaf arrangement, this could be spiral, simple, imbricate. Even in cells, the leaf cells, laminar cells, we have smooth, unipapillate ones, seriate multipapillate. You could see the papilla arranged in a series, and you could see multipapillate ones like these. Look at the alar cells. Look at these, and look at them. The basal enlarged cells that are very prominent in some mosses, but not in all. Position of the Sporogonium divides the mosses into two groups, Acrocarpus and Pleurocarpus. Then the position of the capsule, they could be erect, pendant, horizontal, inclined, nodding. Look at the peristome, unilayered, double, I can show you the next. Haplolepidous, diplolepidous, where there are two rows, one row, and this is typical of Pogonatum. This is annulus, seen in some of the mosses, not in all, which have been close to the annulus that you normally see in the pteridophytes. This helps in the dehiscence of the capsules. The operculum, that's the lid of the capsule, the diversity. You could see prostrate, which is really long, prostelate, comparatively shorter, apiculate, convex, and papillate. Look at the calyptra that protects the developing capsule. Cuculate, mitrate, and pilose, hairy. There are others also, just giving it. And the spores here, smooth and papillose ones. Coming to liverworts, look at the diversity here. This is Barbatus, where all the three ranks of leaves are of the same size. No differentiation. When you come to Legernia species, you find two lateral rows and one smaller ventral row. But when you come to Radula, this ventral row is totally absent. There are others too. Well, in habit, again you have, as in the mosses, erect, prostrate, and pendant ones forms. Look, simple, branched. Even in branching, there are several. Pinnatype, irregular. We have Legernia, Phalaenea. There are others also. Branching is quite confusing at times. Look at the leaf shape. Heteroscyphus has a leaf with teeth, radula, mastigolegernia, corolla. Look at the lobules. This does not have a lobule. In Phalaenea, it's helmet-shaped. Variations, diversity. Coming to the cell type, this is not trigonous, means there is no thickening in the corner. But here you find the corners thickened. Again here. Here you have even intermediate thickenings. Look at them. This is a trigone, this is intermediate thickening. Typical of liverworts. Coming to oil bodies, beautiful. You could see smooth oil bodies, granular. Look at them, segmented. A great cluster type in the segmented. Coming to the periath, this closes, like the calyptra, this protects what? The capsule, the young capsules. Ultimately, the capsule grows out before the dehisces. You could see plain ones, look at the number of plicae, the foldings. Now, similar to the mosses, we have acrogynous, anacrogynous. When the capsule is young, it's within the perianth. When it matures, it comes out. You could see that, how it dehisces. These are the spores and the elaters. Spores, with diversity, you could see them, different shapes, different patterns of the papillae. Elaters, like the teeth that you saw, in the peristome teeth that you saw on the moss capsule mouth, these also help in the dispersal of the spores. But you could see there is a thickening here, but this doesn't. That's a variation. The liverworts, There are two types. They could either be overlapping or forming roses. This is Vixia. This is Stagionia. Look at the air pores, the difference. That's the pore, the number of surrounding cells. This is Cythodium. This is Vibulia. This is Exelmortica. Look at the diversity. Coming to the Amphigasria or the ventral leaves. This is Marcantia. This is Vibulia. Plagiocasma, two different species, one is rupestri, one is intermediate. The position of even the discs. Humoteria has it at the apex. This is Plagiocasma normally has it little behind the apex. Vibulia has it at the apex, and this is Marcantia. This is but male. This is not the female disc, it's a male disc. Come to the spores. There are different types of spores, but I'm just showing you two with prominent differences. Again, in elatus, you find this has a single spiral. This is the elater of Ricardia. You could see the spores attached to it. These are the spores. This is bispiral. You could say two Plagiocasma. Coming to hornworts, because these sporophytes, the simplest sporophytes, but the only thing is that these sporophytes are the ones that resemble the most primitive xylotum of the pteridophytes. So there is a theory that probably from pteridophytes, bryophytes descended through the hornworts. Here the growth is not determinate but indeterminate, particularly. So in the bryophytes, this is some group feel that the sporophyte of hornworts are the most highly evolved ones because of the indeterminate growth of the sporophyte. Now, even there's a Nautotylus. You could see the difference, the arrangement of the sporogonia, the height, the length. This is Anthoceros. Coming to the spores, there's a Phaeoceras spores. You could see the inner face, the outer face. There's the Nautotylus spores. And we come to the elater part, probably the last slide that I would like to show you. In elaters also, when it comes to the onwards, there are two types, pseudoelaters and elaters. Pseudoelaters are more than one cell and they do not have the wall thickening. But when it comes to elaters, elaters are usually unicellular, single cell with spiral thickening. So even there, you find a diversity. Concerning the distribution pattern worldwide, within the continent or within the country, it's best explained by all of us should be familiar with the continental drift theory. That probably best explains the reason for the patchy distribution of these plants, these delicate elusive plants, I would say. Within the country could be because of the Pleistocene glaciation. This is one explanation. However, there is yet another thought that these patchy distribution could be due to inadequate survey and studies. So we have a lot more to study on Indian bryophytes, particularly the southern side, and even worldwide. Still, bryophytes remain to be a little neglected group. That's what I feel, depending on the publication that I see. This could be, I don't say this is the reason, this could be one of the reasons why the distribution is discontinuous. But there are a number of species which are cosmopolitan, distributed worldwide. Well, I think stop with this. Thank you very much for the wonderful opportunity given to me, to Dr. Sanjay.
Okay. Are it through? I mean, completed? Okay. Thank you.
Yes.
And yeah,
Stop sharing.
Dr. Daniels for
Yes.
completing your talk in a very short time. It's good for us for the question and answer. If anyone has any question, want to interact with Dr. Daniels, please put forward your questions and all. Any questions for Dr. Daniel? Switch on your mic and you can speak or raise your hand.
Hello.
Yes, tell me. Yeah.
Sir, okay. Sir, good morning. I am Alan from Palakkad.
Yes.
Okay. Sir, it was a wonderful talk. Sir, I just need a little bit clarification on whether the studies on, is there any scope for further studies on liverworts or hornworts in the Western Ghats or particularly in the southern part?
Is that your question?
Yes, yes,
Yes,
sir.
of course. There are a number of regions, places in the Western Ghats that are awaiting surveys. There are number. So I would never say that my survey and the study is complete.
Okay.
Okay, there are a number of places. You will definitely come across new species, even new genera. Because when I worked in Silent Valley, I have instituted new genera, Indopotia zanderi.
Okay.
So definitely there will be areas, there are a number of areas from which you would get novelties, new reports, etc., etc. This could probably explain why some species distribution remain to be patchy. When we work more, because number of mosses thought to be endemic to the eastern Himalaya and the northeast India, we have reported from the southern Western Ghats. Okay. Similarly, liverworts. Because due to water time, I rushed through. I didn't show you number of my slides because I wasn't sure whether I could take a longer time. So it's that. So there is definitely a lot of scope for
Okay. Sir, just one more question, sir. Sir, there are some leafy liverworts that closely resemble mosses. I could see some of them in your slides also. So, is there any a trick or something so that we can by sight itself we can identify between whether this is a liverwort or a moss in the fields?
See, except for a few species, it is better you don't attempt identification of bryophytes on the field.
Okay.
This is what I have come across. See,
okay.
It is always better not to identify bryophytes in the field. There are some species with striking characters that you could identify on the field. But there are mosses that resemble liverworts also with amphigastria, etc. But the thing is, liverworts, when you look at the rhizoids are always, unicellular. Normally, you don't come across a septa. But in mosses, the rhizoids are normally colored, whereas here in the leafy liverworts, it's hyaline, but in mosses, they are colored, more robust, and they are septate.
Okay.
With this, you can distinguish because there are mosses. Somciella is a good example. In Erpodiaceae family, you get certain species or genera where it is very confusing because they have amphigastria. But this is one way by which, of course, when capsules are produced, if you're able to collect them in the reproductive stage, then it's easy to identify. In case you don't, in the vegetative condition, this is one way of identifying.
Okay.
The cells are also very different. Okay.
Thank you. Happy to be in your class again, sir. Thank you.
Welcome.
Anyone else? There is one question in the comment box by Pooja Maurya. Sir, you mentioned trigonus. What is it? Which type of thickening is there?
Trigones?
Yeah.
Trigones are normally thickening that you find at the junctions of cell walls. a triangular thickening of walls. I couldn't exactly understand what you want to know. Hello?
Yes, you can read that question if you can access to the comment box. You see, sir, in the right there is one dialog box is made. She's asking.
Dialog box.
Okay. Trigonos.
Where?
Pooja Maurya.
Yes, yes. What is it? Say, trigon is a cell thickening. Generally, the cell wall is made of cellulose, as in the case of bryophytes. So trigons are special thickenings of the cell wall of the cells, usually at junctions. So they are usually triangular. Usually, generally. But sometimes between the junctions, you also have thickenings, what you call intermediate thickenings. They are generally cellulosic. Have I made myself clear?
Pooja, are you there? You can switch on your mic and ask, sir. Okay. Now let us go to next question. This is by Abhilesh KS. Sir, the most reliable key to work upon liverworts in Western Ghats.
Most reliable key. A question little difficult to answer. The most reliable key, because I have come out with books carefully studying. Some liverworts we still have not collected in the reproductive state, so that remains a question. So to make a key, most of the keys may be sometimes artificial, applying to the region from where the plants were collected. It's very, very difficult to make keys because of the variability. Not only that, and often you don't collect the plant complete. You get them in the vegetative state. I hope Dr. E.K. Singh, I could see him, would agree with me in making a key.
Daniel, sir.
Yes.
Sir, I'm doing my PhD in Palakkad district. I'm Sachita Menon. My name is Sachita Menon. Sir, is there any relationship with Rixia stricta and Rixia fluitans in habitat, like ecology, like relations is there?
Rixia stricta?
And Rixia fluitans.
Yes. See, fluitans is basically an aquatic form. But as you read, sometimes you find that it also grows in mud, very close to the water source. I've collected both. Okay.
Yes, okay, sir.
Yes, but Rixia stricta is a little different. Though the thalli of both the species look alike, in the spores and the position of the spore.
Okay.
Belgium is different, by which you can make it, because I have collected both Rixia stricta as well as Rixia fluitans.
Okay, sir. Okay, sir. Thank you, sir. A great pleasure to listen to your talk, sir. Thank you, sir.
One question from Manivanan. Sir, how to collection in easy method?
How to collect bryophytes in easy method? What do you mean by easy method?
Shortcut method.
What we normally do, I'll tell you. What we normally do, easy means you can take small polythene bags of 9 by 6. Though polythene bags are not permitted in most of the reserve forests and sanctuaries, but you have to get a special permission saying that these polythene bags will be taken back. They will not be left in the forest as it is, as they are. So you take a marker pen, you have an altimeter, okay, and if you're interested in other parameters like relative humidity, whatever, you could take them along. You have a marker, you quickly note down, collect the sample. You can use a small scalpel to remove, a small knife-like thing to remove, okay, and then put them in the polythene bag and immediately write the elevation, the habitat, and the vegetation type. If you're a good botanist, then what you could do is you can probably identify the host tree also, or take a good botanist along who's much familiar with the angiosperms of the particular locality. What you could do is you could ask him and get the host plant identified too, which we always try to do, but sometimes we fail because when we are in the depth of the forest, at times you cannot see the crown unless you get leaves and flowers at a reachable height. So that this is the way that we do, and come back and then quickly write down in your field notebook all the details. I don't have any other method to suggest. This is what we do, and if you like, you could follow.
Yeah, you need to dry it also, no, sir? You dry it.
You just ask me about the collection.
Okay.
Ask me about the collection, and what I do is sometimes the collections are more. If you can't do them immediately, the identification, we store them in a refrigerator in the lower racks. Then one by one, you can work on them. They're there for nearly 6 months. They remain fresh. You could work as and when you have time to do. And then once you identify, then you could dry and then put them in covers of 15 by 10 centimeters and have all the details on the flap.
Yeah.
It's a standard method of preserving dry flies. Dry method. Just dry them, but do not use any as you do in the angiosperms. Don't keep any heavy material to press them. Don't press them. By pressing them, you spoil if there are capsules, you spoil the reproductive structures. So generally, it's advised not to keep anything heavy on the plant material, but just dry them as they are and keep them in covers with all details.
Okay, thank you, sir. Anything else? Anyone else want to ask?
Daniel sir,
Yes.
Manju,
Yes.
very nice to hear you both. DK Singh sir also.
You spoke yesterday. I
If
think
you are
it's
wonderful. Yeah, yesterday also.
No.
So we are in college. I am sitting there.
Okay.
That's why. So nice to hear both of your talk. Very happy to see you all in
Nice
this
to meet you.
online mode.
Yes.
Okay, so Sanjita and Nishida both are my students.
Oh, you
They
are
also
students.
present. Yeah.
Good.
Okay, very
DK Singh
good.
sir, it's happy to see you, sir.
Yeah.
Okay, thank you very much.
Yes, welcome.
So, if there are no more questions, we can wind up for this talk. I thank Dr. Dilip Daniels for accepting our invitation, and today he's given a nice talk. Thank you very much, sir.
Welcome, sir. Welcome. Thank you.
So we'll take a short break for 15 minutes, and we'll be back by 11:30 for Dr. Robert Luking's talk on lichens. We'll take a short break. Okay. Thank you. Thank you all.
Video close பண்ணிட்டு
I can see the Dr. Robert has logged in. So very good morning. Dr. Robert, good morning.
Morning.
Yeah. Dr. Robert, I have just announced a break for a few minutes, and we'll start by 11:30 as per the schedule.
Hello, good morning to everyone. Good morning, Dr. Robert.
Good morning.
Okay, good morning, Dr. Upreti.
Yeah, good morning. Dr. Sanjeev,
Yes, sir.
I think you announced that presentation. You have already announced it that it will be start by 2 o'clock or for the students.
Yeah. Participants and students and others. Your presentation, oral presentation and poster presentation will be there after lunch. We'll start that by 2 o'clock, we don't have much time for oral and poster presentation. So for the oral presentation, you will be given just 6 minutes, and you have to summarize your work. And for the poster presentation, 5 minutes will be given. And already you people have informed who is going to do the posters and oral presentations. You have that information with you. And by 2 o'clock, we'll start the presentations. Be ready. Thank you.